I think everyone's just joining now. So my name is Elizabeth Openshaw. I'm the NCITA Education and Communication Manager, and it's my pleasure to welcome everyone to this Nuclear Medicine in Oncology training workshop today. So just a couple of housekeeping rules before we start. So firstly, if you can please ensure you are on mute throughout just to minimise any background noise. The event will be recorded and it will be made available on the NCITA website and also on the YouTube channel. And also, if you have any questions, please do post these in the chat box. So we will have two lots of Q&A throughout the workshop, and we'll be going through those questions in the chat. So please do make use of that. So as an overview for NCITA, for anyone who is less familiar, so NCITA stands for the National Cancer Imaging Translational Accelerator. And we are a network of medical imaging experts and scientists across 9 leading cancer imaging institutions across the UK. And we're really building a unified infrastructure for the discovery, validation and adoption of cancer imaging biomarkers into clinical trials and the NHS. So this workshop today has been designed to provide a comprehensive introduction to nuclear medicine in oncology. And this is for researchers, students, really anyone who would like to find more about this area. So I'd like to give a warm welcome to our speakers today. We have 4. So we have Dr Maite Jauregui-Osoro, the NCITA Imaging QA/QC Manager based at Imperial College in London. We have Dr Jennifer Young, who is Research Associate in Translational Radiopharmacy based at King's College London. We have Edith Gallagher, a Research PET-CT Technologist based at the University of Oxford. And then finally, we have Dr Daniel McGowan, who is a Principal Clinical Scientist at the University of Oxford. So you can hopefully see the agenda on the screen, but just as a quick overview, we'll be having 3 presentations to start with by Maite and Jen, followed by a Q&A session, and then we will have a break from about 2:15 until 2:30. And then we'll go into the second part of the workshop with 2 presentations from Edith, followed by Dan. We'll have another chance for a Q&A session, and then we'll wrap up. So I'd firstly like to properly introduce Maite. So Maite completed her PhD in chemistry at the University of Manchester. She then moved to King's College London as a postdoc and soon after obtained an EPSRC fellowship. And Maite has held a research and regulatory position, was a QC manager in the PET Centre at St Thomas's, and was also a trial manager for the first in human clinical trial with a radiopharmaceutical her team developed. In October 2019, she joined NCITA as the QA/QC unit manager and is currently based at Imperial College London. So Maite, I'd like to pass over to you. Thank you.
Thank you very much for the introduction, Elizabeth, and hello everyone. So I am now going to give you a brief introduction to radiation and production of radiotracers using cyclotron facilities. Nuclear medicine uses radioactive pharmaceuticals, also called radiopharmaceuticals or radiotracers, to diagnose, evaluate, and treat diseases. It includes 2 main diagnostic imaging modalities, PET and SPECT, and a treatment modality, molecular radiotherapy. Now, the focus of this workshop will be on the 2 diagnostic imaging modalities, PET and SPECT. So let's see what happens in these 2 modalities. Well, first of all, we need to obtain or produce a radionuclide, which will be the source of radiation. Then we need to incorporate this radionuclide into the drug or pharmaceutical we want to study. And once we've done that, we administer the radioactive pharmaceutical to the patient, and we wait for it to accumulate at a tumor or other target sites and clear from the rest of the body. Then we scan the patient and detect the radiation emitted by the radiopharmaceutical accumulated at the target sites. And finally, we view and analyze the images that will show how the radiopharmaceutical is distributed in the body and where those target sites are located. So as you can see, nuclear medicine involves the use of radiation. And what's radiation? Well, radiation is energy that comes from a source and travels through space and may be able to penetrate various materials. So for example, light, radio, and microwaves are types of radiation, but these are called non-ionizing radiation. Now, the kind of radiation discussed during this session is called ionizing radiation because it can produce ions, charged particles, in matter. So ionizing radiation is produced by unstable atoms. Now, these atoms are different from stable atoms because unstable atoms have excess energy or mass or both. Atoms with unstable nuclei are said to be radioactive. And in order to reach stability, these atoms give off or emit the excess energy or mass. And by doing so, they turn into another element. Now, these emissions are called radiation. And sometimes isotopes decay from one stable isotope into another unstable isotope continuously in a long radioactive chain until finally stability is reached. The kinds of radiation, well, the kinds of ionizing radiation are particulate, where mass is given off, and electromagnetic, which is like light. So gamma radiation and X-rays are examples of electromagnetic radiation. Gamma radiation originates in the nucleus of the atom, while X-rays come from the electrons. And alpha and beta radiation are examples of particulate radiation, both originating in the nucleus of the atom. Alpha particles, due to their charge and mass, they interact very strongly with matter and only travel a few centimeters in air. So they cannot penetrate the outer layer of dead skin cells, but if you ingest an alpha-emitting substance, it can cause serious damage. Alpha-emitting radiopharmaceuticals, for this reason, are used for radionuclide therapy. So what about beta radiation? Well, beta radiation has smaller mass than alpha particles. So beta radiation can travel a few meters in air and can be stopped by a thick piece of plastic. Beta radiation can penetrate the skin a few centimeters, and therefore it can pose a certain degree of external health risk. However, the main threat is still primarily from internal emission of ingested material. And finally, gamma radiation, unlike alpha and beta, it does not consist of any particles. Instead, it consists of a photon of energy, so it's like a light. And as they have no mass or charge, gamma rays can travel much further in air than alpha and beta particles. And gamma radiation can be stopped by a thick or dense enough material such as lead. So during this workshop, we will be focusing on positrons and gamma rays. So how is radioactivity measured? Well, the International System Unit is the becquerel, which represents a rate of disintegration of 1 disintegration per second. So for many years, and still in some countries, radioactivity was expressed in curies, which corresponds to the radiation emitted by 1 gram of radium, which is the radioactive element found by Marie Curie. But due to the high value of 1 curie, microcuries and millicuries were very commonly used. And since then, physicists adopted a more logical unit, which is the becquerel, which, as I said, corresponds to 1 disintegration per second. So the associated inconvenience here is that the becquerel is a very small unit, so megabecquerels and gigabecquerels are commonly used. So here you can see the conversion factor from curies to becquerels. So how can you protect from radiation? Well, the guiding principle of radiation safety is ALARA, which stands for as low as reasonably achievable. This principle means that even if it is a small radiation dose, if receiving it has no direct benefit, then you should try to avoid it. Now, to do this, you can use 3 basic protective measures in radiation safety: time, distance, and shielding. So time simply refers to the amount of time you spend near a radioactive source. And you should minimize your time near the radioactive source to only what it takes to get the job done. So finish as soon as possible and then leave the area, because half the time spent means half the radiation dose received. Distance refers to how close you are to the radioactive source. So you should maximize your distance from the source as much as you can, because distance and dose are inversely related. So if you increase your distance, then you decrease your radiation dose. And finally, to shield yourself from a radiation source, you need to put something between you and that radiation source. So as we have seen before, the most effective shielding will depend on what kind of radiation the source emits. And some radionuclides emit more than one kind of radiation. So this obviously has to be taken into consideration when choosing the shielding. So what happens with PET radionuclides? Well, we said PET radionuclides emit positrons, which can travel a relatively short distance. Now, during this travel, they can meet an electron. And when a positron and an electron collide, they mutually annihilate, and they produce two gamma rays that are emitted in opposite directions. And it is these gamma rays, and not the positrons, that a PET scanner will detect. So these gamma rays have high energy, and they require thick lead for effective shielding. Radionuclides used for SPECT also emit gamma rays, but these are generally lower energy gamma rays than the ones emitted by PET isotopes. And therefore, they do not require that much lead for effective shielding. So for example, while in order to reduce the intensity of radiation of a PET isotope by half, the thickness of the lead needs to be approximately 6 millimeters. For technetium, that thickness is only 0.33 millimeters. So as a consequence of this, in all radiochemistry laboratories, PET centers, or any areas where PET isotopes are used, there is a lot of lead around. And this obviously has cost implications when designing a PET radiochemistry or scanning facility. Similarly, areas where there is radiation will display many signs and warnings about the presence of radiation, and in some cases also indicating the type of radiation, which radioisotope, and how much. These areas are controlled, and entry is restricted to radiation workers. So if you are a radiation worker, you will have personal protective equipment you can use to minimize exposure. You may also have alarming dosimeters to help manage the time and track your accumulated doses in areas with elevated radiation levels. And if you are handling radioactive substances, your hands may receive a high radiation dose, and this needs to be closely monitored. And this can be done using these rings shown in this picture. So if you're a radiation worker or a patient receiving radiation, it is important to monitor the radiation you receive. But how much radiation is too much? Just measuring the amount of radiation you are exposed to isn't that helpful. So instead, we need to know how much harm that radiation can do if it gets into your body, especially if it's ionizing radiation. So yeah, just knowing there is radiation around you isn't that helpful in working out how risky it is. So instead, you need to understand how the different sources and types of radiation cause biological damage. And this is measured using sieverts, which are a measure of the potential biological impact of exposure to ionizing radiation, and in particular, how this exposure might lead to problems by damaging your DNA. So this helps us understand, for example, how much exposure to radiation might increase the chances of developing cancer. So to do this, sieverts combine three aspects of radiation: the amount of energy that radiation is capable of dumping into your body, and this changes with different sources and situations, the type of radiation, and then which part of the body is being exposed. So by combining these three factors, you get a good idea of how much damage the exposure might cause. And with this in mind, those limits are set to protect workers and members of the public from the effects of ionizing radiation. So they are set at a level that balances the risk of exposure with the benefits of using ionizing radiation. And the fundamental requirement is for employers to reduce all exposure to ionizing radiation to as low as a level as possible, as this should be, and this should always be below the dose limits. Now, those limits are intended to reduce the risk of serious effects occurring, such as cancer, and they are in place to protect the eyes, the skin, and also the extremities against other forms of damage. And depending on the dose staff are likely to receive, they may be classified. And if they are classified, then, for example, they will need to have mandatory medical surveillance regularly. But it's also important to put things into perspective. And we are all exposed to natural radiation sources, which come from the Earth, such as rocks and soil, and the outer space, cosmic rays, and the food we eat. A small amount of radioactive material exists naturally in our bodies. And every year, each person is exposed to natural radiation and also a variety of other sources, such as color TV sets, air travel, which increases the exposure to cosmic radiation due to higher altitudes. So here in this list, you can see estimated radiation doses for different activities, such as watching TV, traveling by plane, having a mammogram, just the dose you get by living in the UK every year, what an FDG PET study may give you, and what the tumor receives during a cancer treatment. So due to the use of radiation in nuclear medicine, there are certain regulations that apply. The control of radiation exposure to individuals while at work, including persons exposed as a result of the employer's work activity, is covered by the ionizing radiation regulations. And the main aim of these regulations is to establish a framework for ensuring exposure to ionizing radiation arising from work activities is kept as low as possible without exceeding those limits that I showed you earlier. So health and safety inspectors have statutory powers to assist them in enforcing these regulations. And the Environment Agency ensures that all radioactive waste is managed safely with no acceptable risks to people or the environment. In these pictures here, we can see how different things were before the introduction of regulations, when they still didn't know so much about radiation and no shielding was used here. There was no PPE, so you can see no gloves, direct contact with radiation sources. And in this picture, for example, you can see that operators are wearing full PPE, protective measures and shielding is being used, syringe shields, and so on. So now let's go back to talk about how radiopharmaceuticals are made. I mentioned earlier that in order to make a radiopharmaceutical, we first need to obtain or produce the radioactive source, the radionuclide. So how are radionuclides generated? Well, for most radiopharmaceuticals used in PET, this radioactive source is generated in a cyclotron, which is a compact particle accelerator. Cyclotrons nowadays look like these images here. In some cases, like the cyclotron at the bottom, they need to be placed into a concrete bunker, which is usually about 1 meter thick, with interlocks on the entrance door. But in some other cases, like the cyclotron on this image, they can be self-shielded if they are constructed with a concrete lead mix. Well, and as you can see on this picture here on the right, cyclotrons are not new. The cyclotron was invented in 1931, and Lawrence was awarded the 1939 Nobel Prize in Physics for it. So what does a cyclotron actually do, and how does it create the radionuclides? Well, a cyclotron is not a simple piece of equipment, and it will be seen differently by different people. So here we will try to explain this in a simple way. So we will focus on this middle picture. So the cyclotron body consists of 2 electrodes called Ds because of their shape, this one and this one. So these Ds are in a vacuum chamber, which sits between the poles of a large magnet that creates a magnetic field. So a stream of charged particles is fed into the center of the chamber, and a high-frequency alternating voltage is applied across the electrodes. So this alternating voltage attracts and repels the charged particles, causing them to accelerate. And the magnetic field moves the charged particles in a circular path. So as they gain more energy during the acceleration, they spiral outwards until they reach the outer edge of the chamber. So here, at this outer edge, what we do is we put a non-radioactive isotope. And when this non-radioactive isotope reacts with this stream of charged particles that come from the cyclotron, a nuclear reaction occurs and a radioactive isotope is produced. So this will be the radiation source. So by changing the non-radioactive isotope that you put at the outer edge of the chamber, different nuclear reactions will take place, and therefore different radioisotopes can be produced. So in this example here, for example, they have placed nitrogen at the outer edge of the chamber. So when the protons are accelerated in the cyclotron and they hit this nitrogen, carbon-11, the radionuclide, is produced. Here I have listed the radionuclides that are most commonly used in PET, along with their half-life and some examples of radiopharmaceuticals produced from these radionuclides. So as you can see, most of these radionuclides have very short half-lives. And by the way, half-life is the time it takes for radioactivity to halve. So if we look at this example graph here, F18, for F18, for example, it takes 2 hours for the radioactivity to decay from 1,000 megabecs to 500 megabecs. But for copper-64, it takes 12 hours to do the same. So the shorter the half-life, the faster you need to work to produce and inject a radiopharmaceutical. And it is for this reason that radiopharmaceutical synthesis must be as simple as possible, automated with the use of robotics, and as quick as possible. So here you can see some examples of synthesis modules. Some of these modules use disposable cassettes that are specific for the radiotracer you want to synthesize. So you can buy the cassettes you need along with the reagents or ingredients for the synthesis and connect everything to the synthesis module for the synthesis to happen in an automated way. But as there is a lot of radiation involved during the synthesis process, shielding must be in place to protect the operators. And therefore, those synthesis modules I just showed you in the previous slide must be placed inside hot cells, which are radiation containment chambers like the one you see in this picture. So while the radionuclide is being produced in the cyclotron, the operator prepares the synthesis module, adds the required reagents, and checks everything is ready for the synthesis to happen. So then the operator closes the door of the hot cell, and the radioactivity is delivered from the cyclotron to the synthesis module inside this hot cell. The operator can then follow and control the synthesis through the laptop. And then at the end of the synthesis, the radiopharmaceutical is delivered to a vial that sits in a lead pot. The operator will then dispense the radiopharmaceutical and perform QC checks before it is released for patient use. Radiopharmaceuticals are medicinal products and therefore must be made in line with the UK Human Medicines regulations. The manufacturer must follow the GMP principles, and GMP is an integral part of quality assurance, which ensures that products are consistently produced and controlled to the quality standards appropriate for their use. The basic requirement of GMP is that all manufacturing processes are clearly defined, reviewed, and shown to be consistent, to consistently produce the pharmaceutical of the required quality standards and specifications, and that all critical steps or any significant changes to the procedure are validated. It's for this reason that GMP covers all aspects of manufacturing, from starting materials, equipment, premises, documentation, training, personal hygiene, etc. So how long does the production of a radiotracer take? Well, let's use the synthesis of FDG as an example. So first we need to produce F18 in a cyclotron. This takes about 2 hours. And during this time, the operators will prepare the synthesis module and the reagents. At the end of these 2 hours, let's imagine we have made 70 gigs of F18. We then need to deliver this radioactivity from the cyclotron to the synthesis module in the hot cell, which can take about 15 minutes. And then do the synthesis, which let's say takes 30 minutes. So just because we've spent 45 minutes in this process, our original 70 gigabecquerels have decayed to just 52 gigs. We then dispense the radiotracer and perform QC checks. This can take another 45 minutes. So at the end of this, we could have 39 gigabecquerels. But this is assuming that we have had no losses and that the yield of the synthesis is 100%. Well, in reality, this is lower. And for FDG in particular, this is approximately 70%. So at this point, we can expect to have about 25 gigabecquerels in reality. If the QC checks are satisfactory, then the radiotracer is released for use and can be shipped to the hospital. The scanner can be on site or can be at some distance. So if we need to use a courier to ship the tracer, then we will need to use an appropriate courier and a type A container, like the one shown in this picture here, which protects the contents and provides sufficient shielding during transportation. If the hospital is, let's say, 45 minutes away, then from the original 70 gigabecquerels we started with, we can expect to get about 19 gigabecquerels when it gets to the scanning site. So now I'm going to show you a quick video of what happens once the tracer is injected into the patient. So that was the end of my presentation. So thank you very much for listening. And I will be happy to answer any questions you may have during the Q&A session later on.
Okay, fantastic. Thank you very much, Maite. So we're now going to move on to Dr. Jennifer Young. So as a quick introduction, Jen is a postdoctoral research associate at King's College London, and she has a background in chemistry and completed an MRes in medical imaging at King's College London. This led to a PhD entitled Imaging and Therapeutic Radiotracers for Prostate Cancer. And she gained GMP experience setting up gallium-68 radiopharmaceutical production at St Bartholomew's Hospital in London. She returned to King's College London in February 2020 to join NCITA as the chemistry specialist within the QA/QC team. So thank you very much, and over to you, Jen.
Thank you very much, Elizabeth. Hi, everybody. As Elizabeth said, I'm Jen Young. I'm going to give you an introduction to radiopharmacy today. So here you can see some pictures of the radiopharmacy, and this is where radiotracers are made using aseptic manufacture, which basically means you're trying to ensure that all the products that we're making are sterile. A radiopharmacy is normally within the hospital, and the manufacturing unit has to have lots of controls in place to ensure that everything's sterile. As you can see here, we have specific sterile isolators that have gloves or gauntlets at the front, and it's in these isolators that the radiotracers are made. All staff within the production unit have to gown up, and they also have to cover their hands and their hair, and there's very strict cleaning protocols throughout. So staff working in radiopharmacies, and in fact staff working in PET centres like Maite described as well, tend to be split into 3 teams. The production team, who manufacture the radiotracer, the quality control team, who conduct tests on the radiotracer to ensure it meets specifications, and the quality assurance team, who check all of the documentation and are the final people that release the radiotracer for use. These 3 teams must be independent from each other. This is part of the GMP requirements, and it helps to ensure quality. Something slightly different has to happen if the radiotracer is an investigational medicinal product for use within a clinical trial. In this instance, it must be a qualified person or QP that carries out that release. These people have additional training and certification compared to other members of the QA team, and they have to be named on the IMP license for the radiopharmacy or for the PET centre. So it's really important that you have this person available if you're doing a clinical trial. So in what way is radiopharmacy production different to the type of radiotracers that Maite talked about, which are made at units that have a cyclotron? Well, radiopharmaceuticals that are typically made in the radiopharmacy focus uniquely on radioactive metals. And because these are metals rather than non-metals, they require a different type of chemistry in order to form radiotracers. Metals can easily be incorporated into compounds using specially designed molecules called chelators. The word chelator is derived from the Greek for claw, and we can imagine that there are a number of claws holding the metal in place. In radiotracers, lots of these claws are joined together so that they totally surround the radioactive metal and bind it really tightly so that it's stable when it's been injected into a patient and it gets to the disease site. So how are radiotracers designed? So radiotracers can be made up of just this chelator with the radio metal going into the center, or they can be a bit more complex. You can also have a situation where you have the chelator, which is then attached to a linker, which is then attached to a targeting molecule. So then when you add the radioactive metal, this will then specifically go to a disease cell, which may express a certain type of protein or targeting species on its surface. So in order to understand how radioisotopes are generated within a radiopharmacy, I wanted to introduce you to the concept of a decay scheme, where one radioactive isotope called the mother decays into a second called the daughter, which then decays into a stable element. I want to talk you through this process because it's slightly different to what Maite talked about with cyclotrons, and it forms the basis of how radiometals are generated within a radiopharmacy. So in this scheme, we're really most interested in the daughter isotope. So this is the one in the middle of this scheme, as this is the isotope that's going to be used for imaging. It's the one that's incorporated into the radiotracer. It's got a short radioactive half-life, which minimizes the dose to the patient. The mother isotope is the one that's going to produce that daughter isotope we're interested in. And this one needs to have a longer radioactive half-life on the scale of weeks, and it must be easily separated from the daughter. And then lastly, we have the stable isotope that is no longer radioactive. So here are the decay schemes for the most common radioisotopes used within radiopharmacy. So these are both radiometals, and they are technetium-99M, used for SPECT imaging, and this is its decay scheme. And you can see the one we're interested in is the one in the center. And gallium-68, which is used for PET imaging, but it's produced in radiopharmacies. Again, it's the daughter isotope that we're most interested in. This is used for PET-CT. So the source of the radioisotopes within a radiopharmacy is a generator, which is a pretty simple piece of equipment. You have a column on which you load the mother isotope. Then over time, the mother isotope will decay into the daughter. So you can see the red turning to the green here. And this is the isotope that you want, and you want to be able to separate. So in order to separate this, you flush the column with a solution, which removes just the daughter isotope. And this solution, or generator eluate, so we call it generator eluate, is what is used to manufacture the radiotracer. This generator can stay in the radiopharmacy for many days, and you basically elute the generator when you need to make the radiotracer. So you basically have this daughter isotope on demand. So unlike a cyclotron, these are much smaller pieces of equipment. Here are some examples of a technetium-99m generator and a gallium-68 generator. They're about the size of a shoebox, but they're really heavy as they're shielded with lead. The technetium-99m generator has a 21-day shelf life, so you can keep it in the radiopharmacy and use it to make technetium-99m over that period of time. And the gallium-68 generator has a 1-year shelf life. And this difference is basically due to the properties of the mother isotope. The mother isotope for gallium-68 has a much longer half-life. So that's where those differences come from. So how do we use it within the radiopharmacy? Well, the generator sits in a specific drawer within this sterile isolator, and it can be eluted, as I'm going to show you in the next video. Oh, here's the drawer. So you open up the hatch to access the generator. You take off a cap to reveal a needle, and then you take a shielded evacuated vial, which is in that silver metal thing there, and you put it on top of the generator. The needle that was on top of the generator pierces the vial, and the vacuum inside the vial pulls that liquid from the generator into the vial, and then it's ready to use. So the generator eluent is then added to a kit to form the radiotracer. These can be single vial, 2 vial, or multi-vial kits as seen here. But basically these kits contain everything that's needed for the radio labeling. It contains the chelator that's going to bind that radioactive metal and target it to the site of disease, plus other ingredients such as buffers, filler, antioxidants, reducing agents, bactericides, things like that. So the kind of process that you'd go through in order to make a radiotracer in a radiopharmacy is shown here. So this example is specifically for how you would make gallium-68 dotatate, which is used to image neuroendocrine tumours and is made in a lot of radiopharmacies across the UK. So basically you take 4 mL of water for injection, you add it to this kit, dissolve up all of the solid that's in there. You then add a small amount of buffer and you draw up that solution into a 10 mL syringe. You then elute the generator and then add the contents of the syringe that you had to that eluate from the generator. You then heat it up in a heating block and then it's ready to be used after it's been QC'd. So you remove a QC sample. So it's really important to work quickly and efficiently to produce radiotracers to ensure that there's enough radioactivity left for the patient. As soon as you elute the generator, the radioactive isotope starts decaying. And so as Maite explained, the number of megabecs of radioactivity you have drops over time. So typically you have about half an hour to perform this whole production process. So what happens after you've produced the radiotracer? Well, you need to carry out quality control tests. This section is applicable to both radiotracers made in a PET centre, as Maite described, so ones that originated from a cyclotron, and also ones that have been made in a radiopharmacy where the radioisotope originated from a generator. So QC tests that need to be completed are specific to that radiotracer, and these are defined in the pharmacopoeia monograph, or if it's already got marketing authorization, in the summary of product characteristics for that specific product. You can see a zoom in of some of the instructions there. So I'm going to take you through some example tests. So as I said, not every single test will be done for every single radiotracer, but it just gives you a feel for the type of test that might happen. And if you hear that one of the tests has passed or failed, you'll have an idea about what that means and how we could improve that in the future. So one of the first things we want to do is we want to measure how much radioactivity we've got. And we can do this using a machine here called a Kaplan-Tech or dose reader. You measure the radioactivity level, check that you've got enough for all the patients that you want to use the radiotracer for. You also need to make sure that you've definitely got the right radioactive isotope, and somehow you didn't incorporate any radioactive impurities. And this can be done by checking that the half-life you expect for the radiotracer is the half-life that you measure, or checking all of the emissions from the QC sample. So checking that it's decaying in the way you would expect for the product that you wanted. Also, we need to actually visually inspect the vial before we send it out to be administered. Make sure there's no cracks, that the solution's the right colour, that it's the right volume. And we also need to check that the label is correct. We also need to check that no bacteria is present in the sample. And we do this by adding a small sample of the radiotracer we've produced to a batch of broth, and then waiting to see if bacteria grows. This testing actually takes 2 weeks to provide results. So it actually can't be done before administration to the patient. So instead, we have to show compliance through validation and historical track record. And Maite is going to talk about the specifics of these type of tests that have to be done after administration to the patient. We get the results after administration to the patient in her next talk. So we can actually test for some types of sterility before administration, and endotoxin tests are one of the ways that we can do this. So endotoxins are found in gram-negative bacteria and they are toxic, and so we need to check that they are not present in any radiotracers we produce. We can do this with a machine and a cartridge, as shown in these pictures, and you get a readout to show that it's passed. It's also important to check things like the pH is correct, and this can be done with things like pH paper. One of the most important tests is to check that you've actually made the product that you were expecting, and this can be done using something called radio TLC. So this checks that you have bound all of the radioactive metal into the desired radiotracer. In this example, you basically put a bit of radiotracer onto a special paper, strip of special paper, and then you put it into a tube containing a small amount of solution. This solution moves up the paper, and hopefully the radiotracer will move up the paper if it's also been made correctly. And if it hasn't been made correctly, it will remain at the baseline. And then, because you can't see the radiation with your eyes, you have to use a detector to provide you with a graph of where the radiotracer is located on that strip of paper. And this is the machine that we use for that, and you get a graph that looks something like this. And you can measure the ratios of the different products on the graph. A similar test that you can use to check for purity is radio HPLC. This is basically trying to do the same thing, but it's able to distinguish between more compounds at one time. So it gives you more information than your ITLC can. So the way that this works is that you inject the radiotracer into the system and it travels to a column. And different compounds take different amounts of time to be released from a column. And so this is how they're separated from one another. Once the radiotracer leaves the column and goes through the detectors and then into waste, you get a graph showing you at what time the radiotracer came off the column, and you can check that every time you do the synthesis, you get the same graph, and you can also check that this agrees with a known standard as well. So you're expecting to get nice graphs that correspond to your standard and your radio tracer. Another test that we might need to do is filter integrity. And this machine is used to check that any filters that might be used in the process in order to sterile filter the radio tracer and remove any possible bacteria were actually working at the time that they were used and that they didn't have any leaks. And this basically confirms that the product is sterile. So this is a range of different tests that might need to be done on a radio tracer. And again, it is super important to do these QC tests quickly and effectively. You typically have about half an hour to perform all of these tests, and you need to do that as soon as it comes out of production. So the next step is quality assurance, checking that all of the steps of production and QC have been done correctly. All the tests have passed, everything was in date, everything was labelled correctly. And so you're going to look through the production report and the QC report, and then put together a release document. So for non-clinical trial radiotracers, this can be done by any member of the QA team. But again, specifically for IMPs, so for investigational medicinal products of clinical trials, it has to be a QP that signs that release document. The other thing that the other document that the QA release might also cover is the transport documents as well. They're likely to be responsible for checking these as there's lots of regulations around transport as well. But as I said, again, it's very important that this part of the process happens in a timely manner. So it's really important that you ensure that the QA or the QP is available at the required time to sign off the radio tracer. It's particularly important for QPs because nobody can substitute in for them, and QPs are often based across many sites. So you need to ensure that they're available at the required time, at the required site for the radio tracer manufacturing when you're doing a clinical trial. So you then put the radio tracer into a case ready for transport into a patient, transport to a patient, and this might be within the hospital or it might be at another site. So as I said, it's important to check everything's labelled correctly and you're complying with all the transport regulations. So again, this is really important to be done on time and efficiently. You want to ensure the courier is going to arrive on time and take the product directly to the site, as any delays could mean the radio tracer can't be administered to the patient. So if everything goes well, then the tracer will get from production to the patient in about an hour. And this requires a lot of teamwork and coordination. But unfortunately, sometimes things don't go as planned. So I'm just going to talk to you about, unfortunately, failures now. So sometimes radio tracer production can fail. Such failures actually happen in the manufacture of any GMP products. But if you were talking about a non-radioactive drug, you tend to have a much longer expiry. And so you can store up a stock of product. Which means that if you're doing a clinical trial or if you're working in the hospital, you're rarely going to see the impact of these production failures because there's enough stock to cover. However, when we're talking about radio tracers, these have a really short half-life, which means that they've got a really short expiry time from being made. And so they're basically made on demand for a patient. And so, of course, if a failure happens, then it's going to have more impact. So a failure might be detected at any part of the process that I've described. It could be during production, it could be QC or QA. There could also be a problem in transport. And if the product isn't fit for purpose, then it wouldn't be released for administration to the patient. So the QA or the QP wouldn't be able to sign it off to say, yes, you can go ahead and administer. It's likely that if a failure does occur, a patient will have to be rescheduled, and this is going to be covered by Edith in her talk, how best to think about rescheduling patients and the implications if you're doing different types of studies. And so I just wanted to explain why it's not often possible to repeat the synthesis on the same day. So say the radio tracer failed at 10 a.m. Sometimes it's not possible to repeat it in the afternoon. And this is because of the setup and manufacturing time within the radiopharmacy. It's also due to the staffing hours, and I'm going to talk to you about that on my next slide. And also just kind of physical things in that the generators I've explained about earlier in my talk, it takes about 2 to 6 hours to regenerate enough technetium-99m or gallium-68 in order to be able to make another batch for a patient. So you kind of got to wait that period of time anyway before you can even have a chance of making it again correctly. If the radio tracers made from a cyclotron produce radioisotope, then you need to ensure that there is time on the cyclotron in order to produce that. And also you need to make sure that by the time you've made the product again, that there's actually enough time left in the day to image the patient. So just to give you a little bit more insight into why it might be hard to repeat a production run on the same day, or why you would need to give a radiopharmacy appropriate notice if you decided you have to change when a radio tracer needs to be produced, I wanted to give you an overview of a day in the radiopharmacy. So as you can see, the start time is very early. Staff in the radiopharmacy don't work 9 to 5, but typically 6 till 2. And this, of course, was going to impact on when you might be able to talk to a member of radiopharmacy staff, and also how far in advance changes need to be made. So normally the technetium-99m production, which is the bulk of the radiopharmaceuticals that a radiopharmacy tends to produce, is going to happen early in the morning so that radio tracers can be available and sent to nuclear medicine departments so that patients can start to be administered and then scanned from 9 a.m. onwards. Due to the shorter half-life of gallium-68, gallium-68 radio tracers tend to be made later in the morning, and it is most likely that it's going to be that gallium-68 slot, which is the one that will change if you need to do additional runs for a clinical study or clinical trial. So you can imagine if you're already doing it much later in the working day, then if it goes wrong, you're not going to have enough time to reschedule it with all the staff that you need. And as I said, you need all members of those 3 different teams, the production team, the QC team, and the QA team to be available to do it. So I just really wanted to give you that overview of what it's like when people are working in the radiopharmacy. So I hope that this talk has provided you with insight into the processes that go on in the radiopharmacy in order to produce radio tracers. And I thought it'd be nice to end with this short video, which recaps the processes that I've discussed with you. So hopefully you'll be able to hear the music that goes along with it too. So yeah, thank you very much for listening, and I'll hand over to Elizabeth.
Great, thank you so much, Jen. Okay, so we're going to move back to Maite, who will be talking about peculiarities of radiopharmaceuticals and consequences for clinical studies. And just a reminder that after Maite's talk, we'll have a Q&A session. So if you have any questions at all, please post them in the chat box. Thank you. Over to you, Maite.
Can you see a presentation?
Yeah, it's showing. Thank you.
Thank you. So hello again. So as you may have seen by now, radiopharmaceuticals have certain peculiarities that make them different from other drugs. So during this presentation, I want to focus on these differences and the implications they may have when conducting clinical studies. So why are radiopharmaceuticals different from other drugs? Well, there are two main differences that I want to highlight. First of all, radiopharmaceuticals are injected in very low concentrations. And secondly, due to their radioactive nature, they only exist for a limited period of time, which is dependent on the half-life of the radionuclide present in that radiopharmaceutical. Now, let's focus on the first aspect, the low concentration. Radiopharmaceutical development shares similarities with drug discovery, but there are key differences that allow a faster, safer, and more cost-effective route for taking radiotracers into human studies. One main difference is that in PET and SPECT imaging, the dose or the concentration of the administered radiotracer is always below the pharmacologically active dose and must never produce a biological response. So this is called microdosing, where the administered dose is below 100 micrograms with no pharmacological effects. As a result, PET or SPECT radiopharmaceuticals only require very basic toxicological assessment before progressing into first-in-human studies. So this significantly shortens development time and the costs, because toxicology studies can be very expensive. They are generally around 100K. So due to this microdosing, the regulatory authorities have cut down the requirements for preclinical safety testing. However, this does not actually mean that radiotracer development is cheap, because as you've seen before, radiotracers need to be produced to GMP standards and require specialist equipment such as cyclotron and radiochemistry laboratories. As they are injected in very low concentrations, the side effects of PET and SPECT tracers are very uncommon, and the concern of toxicity is minimized. Besides, certain aspects that are very important for other drugs, such as oral bioavailability and therapeutic window, do not apply to PET and SPECT tracers. Now, let's go back to how tracers are made. So you will remember from the previous talks that the first thing that we need to do is obtain the radioactive source, the radionuclide. The concentration of this radionuclide ranges from nano to picomolar. So as you can see, it's very, very low. We then mix this radionuclide with the molecule we want to radiolabel, which is called the precursor, and it's represented by the blue circles here. And we add any other required reagents and excipients represented by the green and the gray circles in this picture. So all these components are in much higher concentration than the radionuclide, so they are generally in the millimolar range. So what's the consequence of this? Well, as the concentration of the blue precursor circles is higher than that of the radionuclide, at the end of the reaction, only a few blue circles will be radiolabeled with the radionuclide, and the rest will remain not labeled. Now, in some cases, this whole mixture is injected into the patient. So all these components are injected. And this is generally the case for radiotracers made from generator-produced radionuclides and kits, like Jen explained previously in her presentation. But for radiotracers produced in a cyclotron, there is generally a purification step to get rid of any unlabeled molecules and other unwanted reagents as part of the synthesis. So you end up with something more like this. Now, regardless of which option is injected into the patient, either this one or this one, you can see that the concentration of the radio-labeled molecule is always very, very low. It's in the nano to picomolar range. So what consequences or implications does this low concentration have? Well, firstly, as I have already said, these tracers do not produce pharmacological effects. But also, due to their low concentration, radiopharmaceuticals cannot be tested or characterized or analyzed using standard techniques that would be used to characterize any other drugs. For example, you cannot use NMR spectroscopy or mass spectrometry, because these techniques, even though they can determine the structure of the molecule, they require higher concentration of the molecule, which is what we don't have with radiotracers. So, as we've seen in the previous slide, the drug substance, that is the active ingredient or the radio-labeled molecule, is not isolated from the drug product, from the whole formulation that is going to be injected. So this also makes the characterization of the radio-labeled compound more difficult. So this is why the drug substance is never tested on its own. It is the final formulation to be injected, the drug product, which gets tested. So if you are using a radiopharmaceutical as an IMP in a clinical trial, so you will have an IMPD, an IMP dossier, which is a document that summarizes information related to the quality, manufacture, and control of the radiotracer. Now, IMPDs generally have a defined structure, and this document has separate sections for the drug substance and the drug product. But for radiotracers, the section regarding the characterization of the drug substance, this one, will be pretty empty and mainly will just refer to the section of the characterization of the drug product, because this is what gets tested. So how do we check the identity and purity of the active ingredient within the final formulation? Well, we do this by comparison with the non-radioactive equivalent of the radiotracer. So if our radiotracer is compound X labeled with F18, then the non-radioactive equivalent would be compound X with the non-radioactive fluorine-19 in place of the F18. Similarly, if the tracer is compound X with radio-labeled with carbon-11, then the non-radioactive equivalent would be compound X, but with the non-radioactive carbon-12 in place of the carbon-11. So as opposed to the radioactive compound, the non-radioactive equivalent compounds can be purchased or synthesized in higher quantities, so they can be fully characterized using techniques non-radioactive compounds would normally use, such as NMR or mass spectrometry. Then, after we have done this, using HPLC, we see how the compounds in the drug product behave compared to the non-radioactive reference. So the HPLC will display two measurements, one being the absorbance or conductivity, and the other one being the radioactive measurement. Now, the non-radioactive reference standard will only show results for absorbance or conductivity, but not for radioactivity. In the radioactivity window, we will just see noise, but in the other window, we will see a signal. So here we can see that the reference standard shows a retention time of about 26 minutes. So in theory, our radioactive compound should have the same retention time. So when we test our formulation, the drug product, we can see that the non-radioactive chromatogram shows various peaks here, which will correspond to the other reagents or excipients present in the formulation. Because remember that the concentration of the radioactive compound is so small that will not show here. But in the radioactivity window, however, we see there is something radioactive that has a retention time of about 26 minutes, as expected. So this is good. So finally, what we do is we mix the drug product and the reference standard, and then we test this mixture. So here we can see that both the radioactive compound and the non-radioactive reference standard show the same retention time. So here, with this, we can be pretty confident that the structure of our radioactive molecule is correct. So as we've said before, due to their short half-life, radiopharmaceuticals must be manufactured on the day of administration. Manufacture is not always simple, and failure to meet the required specifications is not rare. Apart from potential hazards of radiopharmaceutical production, the short half-life forces QC procedures to be performed very quickly. Therefore, Annex 3 of the GMP guidelines allow the release of radiopharmaceuticals before completing all QC tests, and it allows radiopharmaceuticals to be administered to patients prior to performing sterility testing. So, radiotracer production occurs in a non-sterile environment in which microbial contamination is controlled and minimized through adherence to strict operating procedures. And the sterility of the pharmaceutical is ensured by filtration of the drug product through a 0.2 micron filter. However, the integrity of this filter, which is assessed after the filtration has happened, is not always tested before the radiopharmaceutical is injected to the patient either. Nowadays, in most cases, the filter integrity test is done in an automated way, and therefore the results are available before releasing the tracer. However, in laboratories where this is tested manually, the results may not be available before release due to the need to protect the operators from receiving a high radiation finger dose by performing the test. Before the tracer is released, as Jen mentioned in her presentation, radiopharmaceuticals are tested for endotoxins, which can cause a systemic inflammation response. However, the injection of a product, the sterility of which is not confirmed, is always far from ideal. So in these cases, as specified in Annex 17 of the GMP guidelines, the manufacturing process of the radiotracer must be validated to prove that the required specifications of the final product, including sterility, can be achieved consistently. As the filter used for the filtration of the tracer is the only mechanism in place to make the final product sterile, it is very important that process control and risk mitigation measures are implemented during the manufacturing process. Now, some of these measures include testing the bioburden throughout the manufacturing process and minimizing the microbial load to the sterility filter that is used during the production. But despite these risk mitigation measures, sterility failure cannot be ruled out prior to tracer administration. And this obviously can have serious consequences because most of these patients are also immunocompromised. On the other hand, any failure to meet any of the pre-release specifications would result in the tracer not being released for patient use. And although this failure cannot be anticipated, at least it is easily picked up before any harm is caused to a patient. As radiotracers are prepared right before their administration, patients will have already arrived to the nuclear medicine department before the end of the synthesis, and therefore before any QC checks are carried out. So consequently, QC failures would cause the inconvenience to the patient who have traveled to the hospital and have the scan cancelled or rescheduled as a consequence of the QC failure. So when such failures happen, the research team must be open and transparent with patients and explain that these things unfortunately do happen. Obviously, any QC failure should trigger an investigation to identify the root cause of the failure and the implementation of corrective and preventative actions where applicable. And in cases where there has been a pre-release QC failure, it may not always be possible to repeat the synthesis. So let's use the example of a PET radiotracer, which, as we have seen before, is synthesized using a module that sits inside a hot cell. So at the end of the synthesis, this synthesis module will be very radioactive. So the operator cannot just open the hot cell, clean the synthesis module, replace the reagents, and prepare it for the next synthesis. Because usually you need to wait about 10 half-lives before opening that hot cell, depending on the amount of radioactivity you used in the synthesis. So if this is an F18 tracer, which has a 2-hour half-life, this means that you cannot open the hot cell until the next day. So if there is a tracer failure, then you cannot repeat the synthesis on the same day. Now, let's use a practical example of the manufacture of a radiopharmaceutical IMP used in a clinical trial. So in this case, the tracer is F18 labeled tetrafluoroborate, PF4. So after the synthesis, pre-release QC checks will be carried out and a certificate of analysis will be issued, like this one you see here. This certificate of analysis will list the QC tests required for this tracer, the release specifications, and the test results. So as you can see here, the table shows the results of all the tests except those of the sterility testing and the filter integrity testing, which in this case was being done manually the day after release to protect the operators from receiving a high radiation dose. So the results will be checked by QC personnel, and in this case, as the radiotracer is an IMP, also by a QP. The QP will also sign a release certificate, but given that not all QC test results are available at this stage, this release certificate signed at this stage is only a conditional certification of the radiotracer. So this document will contain information such as the radioactivity in the released vial, the time of release, the expiry time, and the maximum volume that can be injected to the patient. The document will also state that the batch is being released following satisfactory completion of the first stage QC testing and still pending results of the filter integrity testing and sterility. This is why this document is part 1 of 2. So if all the pre-release tests are satisfactory, the tracer will be injected to the patient. Now, the following day, the radioactivity in the filter used to make that radiotracer sterile will have decayed to safe levels for operators to test it. So they will do the test. So what happens if these filter integrity test results fail to meet the specifications? Well, if you suspect you've done the test wrong, you may be able to repeat the test. But if the test fails again, then you can be pretty confident that the integrity of the filter has been compromised. Now, this increases the risk of the radiotracer being non-sterile when it was injected to the patient. So you now need to wait to receive the sterility results, which take approximately 4 weeks. So in the meantime, a deviation should be raised for the filter integrity test failure. And if the sterility results are fine, then filter integrity failure, even though it will need to be investigated, is not likely to have caused any issues to the patient. But what if the sterility results fail to meet the specifications and show that the tracer was not sterile? Well, if both the filter integrity and the sterility tests fail to meet the specifications, then it is most likely that it is a true failure and the tracer was not sterile at the time of administration. Therefore, a deviation must be raised and an investigation must be initiated to find out the root cause of the failure and implement any appropriate actions. So the results must be reported to the CTU and the sponsor, and then we need to find out if there have been any adverse events or adverse reactions reported. Besides, manufacturing of that tracer for human administration will have to stop until 1 or more synthesis test runs are done, showing that all QC specifications can be met. Now, on the other hand, if the filter integrity test has passed, but the sterility results fail, then we have to first question if this is a true failure or if there were any issues when performing the sterility test. Therefore, the first thing to do is to carry out an investigation about the test. So this will include aspects such as, was there an obvious error during testing, such as testing equipment failure, a power outage, or something similar. But regardless of the testing error being identified or not, there generally isn't any leftover sample to repeat the test. Therefore, we should always proceed as if the result is a true failure and do all those things that I just mentioned earlier, such as report the failure to the CTU and the sponsor, find out if there have been any adverse events or reactions, etc. So when the post-release QC tests are completed, the QP will sign the second part of the release certification. And this will be approximately 1 month after the injection of the radiotracer to the patient. And to finish the presentation, I just want to mention the differences when manufacturing radiotracers as IMPs for clinical trials or as non-IMPs for other clinical studies or routine care. The Medicines for Human Use Clinical Trials regulations require that all IMPs are manufactured according to GMP at sites that hold an MIA IMP license, and that the IMP batches are released by a qualified person. But the majority of the radiotracers used within the NHS are not IMPs used for clinical trials. Instead, they are mainly manufactured within NHS radiochemistry and radiopharmacy facilities under a specials license following Regulation 167 of the Human Medicines Regulations 2012. A specials license is needed to manufacture medicinal products without a marketing authorization, that is, products that are not licensed. So there are certain restrictions that come with products made under a manufacturer's specials license, such as, for example, products must be used for clinical special needs of an individual patient as determined by the prescriber, and you cannot advertise or market these products. So as explained in the MHRA guidance, a qualified person is not required for the release of an unlicensed product under a specials license, and therefore most hospitals do not have the services of a QP permanently at their disposal. So again, this is one of the reasons why not every site within the NHS can manufacture radiotracers as IMPs for clinical trials. So this is now the end of my presentation. So if you have any questions, then I'll be happy to answer during the Q&A session.
Okay, fantastic. Thank you, Maite. So we have a couple of questions in the chat. So if we go through them quickly and then we'll have a break. So the first one is from Chris, and it was about a QP, a qualified person. So who is a qualified person and what makes them a QP? This might be a question for Jen. It came up during your presentation.
Yeah, no, I mean, either me or Maite can answer that one, I think. So a QP is somebody that has specifically taken on an additional qualification, and they have to be a pharmacist or a chemist by background. They have to have lots of experience working in QA and QC, and then they have to demonstrate that experience and do a viva to get the qualification. So it's a specific additional qualification that you need to become a QP, and it basically means that you can then release products that are IMPs, but you get that qualification to be able to release any type of product as an IMP, not specifically radiotracers, but you have to be able, if you want to be on the license for a facility that that makes radiotracers, then you have to show that you have appropriate experience in the specifics of radiotracers as well. So I think that that's one of the reasons why particularly radiopharmacies and PET centres find it difficult to always find a QP, because not only do you need somebody with this special qualification, but you also need someone with experience in radiopharmacy. Maite, do you want to follow that up? Yeah, so as part of, in order for you to be a QP, you need to have experience in a site that has an appropriate license. And as Jen said, so not many NHS sites have those licenses. Therefore, if you're working in a site that doesn't have the appropriate license, then you don't stand a chance of becoming a QP. So there is a huge shortage of QPs in the UK, and obviously pharma companies pay a lot for QPs. So whenever people do become QPs, they generally tend to leave the NHS and get a job elsewhere where they get more money. So I think it's also an important note to note as well that when you apply to have a license that allows your radiopharmacy or your PET centre to produce IMPs, so you have to apply to the MHRA to get a license to allow you to produce IMPs. You have to name your QP on that license, and so you have to know who it's going to be as well, and it has to be someone that's familiar with that site.
Yes, you can have a contract QP as well, but again, you still have to name that contract QP.
Yeah, yes. Exactly. So if you want to produce investigational medicinal products, you both need the license and you need the QP. So it is important for clinical trials. Does that answer the question?
I think it does, but please do. Yeah. Please do mention.
I gave you a thumbs up. It does.
Okay, great. Thanks.
Fantastic. Another question is from Penny. So how many patients can a radiopharmacy typically provide tracer for per day? And how many people does it take to produce one dose?
Okay, so it really depends on whether we're talking about a radiopharmaceutical for a clinical trial or a clinical study, or if we're talking about the kind of general everyday radiotracers. So the kits that I talked about, which work with technetium-99m, you can make lots of those kits in the morning, and each kit can be used for, say, 10 to 20 patients, depending on how much radioactivity you put in them. So in the kind of the bulk manufacturing part of the radiopharmacy, one radiopharmacy can produce enough tracer for 50, 100 patients in a day. But when it's something that's specific for a clinical trial or a clinical study, you tend to do just one run per patient. And it's also particularly important for things that have got shorter half-lives. So for example, the gallium-68 radiotracers that I mentioned, the generator doesn't produce enough activity for more than 2 patients per manufacturing run. So that's a big difference between technetium and gallium. Technetium, the generator can produce loads of activity, but it's a very small amount for gallium. So I would say if you're going to do it specifically for a clinical trial or for a clinical study, it's likely that you're going to do one run per patient.
I think it's meant to be single dose. They have to be per patient, so you cannot do.
Okay. Yeah. So that's what I was going to say as well. It also depends whether you're allowed to split that dose. So the ones that we do regularly and bulk, you're allowed to split it in for lots of patients. You make one kit, you can split it. But normally for clinical studies and clinical trials, as Maite said, definitely for clinical trials, you're not allowed to split that batch up. In terms of how many people does it take, the minimum number of people that you would need is somebody to actually physically manufacture the product, somebody to watch that being manufactured, so like a supervisor, and check that every step's happening correctly. You would need a QC person, and you would need a QA person. So I would say the minimum that I've ever done is 4 people, but there is normally more than that involved in the process. Would you agree, Maite?
Yes, and if it's an IMP, generally you also need a second person checking every step in manufacturing, so that adds another extra person apart from the QP.
Yeah.
And when it comes to tracers made from a cyclotron, well, how many patients you can do depends on how much activity you can generate. So you can run the cyclotron for a longer time in order to start with more radioactivity. So that will help, but depending on what reaction you want to do for whatever radiopharmaceutical you are trying to generate. And the synthesis, maybe it doesn't really matter if you start with more radiation because the yield of the synthesis is low. So you are also limited by how good that synthesis is.
So it's a lot of people you need to be around and a lot of people that you need to coordinate.
Okay, fantastic, thank you. One other question is, how big are the needles you use to inject tracers, and how long does it take to inject? So is it the same sort of time as, say, a vaccine, or as long as a dentist injection?
Could we invite Edith in to answer that one, because she's or we could save it for the end, for the next Q&A. She's probably the best person to answer. I mean, I could, but I don't have any experience in injecting.
It's usually pretty quick, as long as the patient has relatively decent veins. It's usually done through a butterfly or a small cannula. I mean, nuclear medicine tests are used on pediatrics. We do them on children and babies as well. So, you know, some of the tiny little needles we can use even on adult patients if they have very bad veins, which can happen in oncology because, especially if they're on chemotherapy treatment, their veins are sometimes very, very difficult to get into. So it's usually pretty quick. Thanks, Chris.
Okay, and just one more question before the break, and that's from Julia Marcus. So, how are mother isotopes sourced?
Oh yeah, I realised I didn't answer this part of the process. So their mother isotopes are normally made in nuclear reactors, and then those are used to generate the generators. And then it's the generator that is shipped to the hospital. Yeah, they're normally made very centrally. So there's a few places across Europe that can make the generators, and they're associated with nuclear reactors.
Okay, fantastic, thank you very much. Okay, so we'll have a break now, and we're just slightly over time, so we'll have a break from now until 14:40 when we'll carry on with the next talk. So please do grab yourself a drink, and we'll see you back in about 10 minutes. Thank you. Welcome back, everyone. I hope everyone had a nice break. So I'll be passing over to Edith in a moment, just as a quick introduction. So Edith Gallagher is a research PET-CT technologist, and she works in the NCITA Imaging QA QC unit. She's been a nuclear medicine technologist since 2005, and in the past few years, she has worked at the Royal Surrey to run research trials, mostly in nuclear medicine therapies. Last September, she moved to the University of Oxford, where she's been working with the NCITA QA QC unit. So thank you very much, Edith, and over to you.
Thank you very much. Thank you, Elizabeth. It's great to be here. Very excited. For my talk today, I will explain a little about the cameras that we use in nuclear medicine and PET-CT. I'll explain what happens in the department before any patients arrive, also what happens with the patient, and a small example of some images. And finally, the logistics and hurdles when looking at scheduling a scan as part of a research study. But first, what exactly is nuclear medicine and PET-CT? Well, you've heard a little bit both from Maite and Jen. The most basic principle is that a radiopharmaceutical is administered or given to a patient. This can be inhaled or injected or ingested even. And then in almost all cases, we want to see what is happening to that radiopharmaceutical inside the patient. For example, where it has accumulated, and we can do this using a gamma camera. This is an example of some of the tests that would be carried out in nuclear medicine departments. As you can see, there is quite a wide range of the body's systems and organs covered. We do have a very small number of studies where we don't image the patient. Instead, we would take blood or urine samples at various time intervals after giving a radiopharmaceutical and measure the amount of radiation in each of the samples. I should also say that some hospitals have both nuclear medicine and PET-CT in the same department, and they come under the title nuclear medicine. And in other hospitals, they are separate. So apologies if I sometimes group the two together under one term, nuclear medicine. In relation to oncology, these are some of the areas that would have imaging performed in nuclear medicine and PET-CT, and I will go through a few of these in a little more detail later in my talk. So how are we different to other imaging that takes place? Well, an X-ray or a CT scan sends radiation from a radiation source to a detector on the other side of the patient, whereas in nuclear medicine, the patient is the radioactive source, and the camera is detecting where it is inside the patient. And this means that as the patient is the radioactive source, everything that comes out of the patient is radioactive too. Let me explain about the cameras that are used in nuclear medicine and PET-CT. On the left is a gamma camera, on the right is a PET-CT camera. The gamma camera has two heads, which are the two box-like structures in front of the donut. These can rotate and be moved into a variety of configurations. Originally, gamma cameras did not come equipped with a CT component. However, this has changed, and over the last 20 years or so, more and more gamma cameras are installed with a CT, even though the day-to-day working name has stayed as gamma camera. On this gamma camera on the left, the CT is the small grey box in the 12 o'clock position on the donut. The PET CT camera on the left is a long, large donut which has a ring of radiation detectors and a CT scanner all incorporated into the one machine. Each head of the gamma camera is made up of a collimator, a sheet of scintillation crystal, a light guide material, and photomultiplier tubes. The collimator is a thick lead sheet with holes in it, as shown in the right-hand image. As radiation comes out of the patient in all directions, the collimator only allows the gamma photons traveling in a particular direction through to the crystal. The gamma photon reacts with the crystal to give off a tiny flash of light, which is seen by the photomultiplier tubes, and over time, all the light flashes that have occurred in the crystal build up to create an image. Just to recap what Maite previously described, PET stands for positron emission tomography, which is exactly as it says. The radioactivity that is used emits a positron. This positron annihilates with an electron, which results in two gamma photons that travel in opposite directions to each other, which we take an image or tomograph of. For this reason, instead of having a head like a gamma camera, the PET camera is made up of individual crystals arranged in a ring around the patient. And as the crystals are separated from each other, it also means that there's no collimator required. With each of the detections of the gamma photons, some very fancy computer work is able to map back where the annihilation that led to that pair of photons has occurred in the body. On the left, you can see a schematic of how the CT is built into the camera. And as a CT of the patient is taken at the same time, the two images are able to be fused together. It is very important that the patient remains as still as possible for the duration of the imaging. Otherwise, significant movement will cause difficulty fusing the two images together, or at worst, parts or all of the scan would have to be repeated. I'll show a few images later on in the talk. Now let me move on to what happens in the department. Most departments will start very early in the morning to get the cameras up and running. The daily QC of a gamma camera and a PET CT camera can take about half an hour to 45 minutes. And some days we'll have additional weekly tests carried out as well. And it's very important that all the QC is passed prior to injecting the first patient. The gamma camera will have a daily flood performed, it's called. The flood is made up of a uniform sheet of cobalt-57 in a hard plastic covering with no blemishes or bubbles in it. This means that if there is anything odd on the image, it is something going on with the camera. You must be really careful not to drop a flood, as you don't want to do any damage to it or the cobalt to leak out. This is a picture of what a normal flood would look like. It should be nice and uniform in appearance with no marks or anything. Not like any of these images, which is not what any technologist wants to see first thing in the morning. For the PET CT camera, this is what a QC image would look like. You can see all the individual crystals. Again, the appearance of them is very uniform, which is a good sign. Not like this image. Here is an example of what it shouldn't look like. Again, you do not want to see this when you're preparing for a day's list of patients. The dose calibrator is checked every morning using a cesium source. During the day, the dose calibrator is used for measuring all the doses that will be given to every patient. So it is very important that it is working accurately and correctly. When the radiopharmaceuticals arrive in the department, they are all measured and checked for a number of reasons, to make sure that there is sufficient activity for the patients that will be attending, but also to keep a track of how much radioactivity is being held on site at any time, how much has been administered to patients, and how much has not been used and therefore going to waste. Dan McGowan will explain a little more about this later in his talk. And sometimes accidents happen. This is a vial that was recently delivered to a department which was discovered to have cracked in transit. It meant that a number of patients had to be rescheduled for another day, which was unfortunate as the patients had to fast for a minimum of 4 hours prior to arriving, and some had travelled quite a distance. To summarise some of the differences between nuclear medicine and PET, you can see that the energy of the radioactivity used in general nuclear medicine is much less than in PET, which is 511 keV. And there are significant differences in the length of half-lives. PET is usually always measured in minutes, whereas nuclear medicine is measured in hours, and some is even measured in days. I've talked a lot about the equipment that we use, but all of this is nothing without our patients. So I'll now go through in general what happens to a patient when they come through to the department. After the patient arrives, they will usually have a short chat with one of the imaging staff. This is to make sure that they understand why they have been asked to attend and to explain what will happen, to check that they have followed any particular preparation required if there's any for that particular test. And to find out if they've had any injuries or surgery or previous cancer treatment, as this could affect their imaging. For example, if a patient is attending for a bone scan and says that they recently fell in the bath and bruised their chest and ribs badly, then if the radiologist sees anything in that area, it could be attributed to that injury. And obviously you want to know in advance if your patient is likely to faint when you go to inject them. Administration of radioactivity to a patient is a highly regulated area. The person who refers the patient for the scan must be a registered healthcare professional as defined in IRMA regulations. This referral is then justified and authorized by a licensed practitioner under RSAC. For novel research radiopharmaceuticals, it may be necessary to update the practitioner's RSAC license. Dan McGowan will explain about the different regulations in a little more detail in his talk later. As a technologist, our checklist is that the referral is justified and authorized by the licensed RSAC practitioner, that we have the right patient, we have the correct pharmaceutical, the correct dose for the correct procedure for the clinical question. This is some of the equipment that would be in a nuclear medicine and PET-CT departments for drawing up the radiopharmaceutical injections and the disposal of syringes afterwards. Due to the weight of shielding required for a vial in PET-CT, automatic dose dispensers are used in many departments. I'm sure you can appreciate that the weights of the vial shields, syringe shields, and the syringe carriers are quite significant when being used by staff multiple times a day, every day. All staff that work in nuclear medicine and PET-CT are monitored to check how much radioactive exposure they are receiving. One method of doing this is by using finger monitors. On the left, you can see 2 finger monitors being worn, one held at the top of a plastic sleeve over the finger and the other in a ring at the base of the finger. These would be worn underneath gloves, and it has a noticeable impact on finger dexterity. On the right, you can see a simulation of a radioactive injection. This requires a certain amount of skill, as the shielded syringe containing the radiopharmaceutical can be quite tricky to attach and detach from the cannula due to its weight, and to make sure that the radioactivity doesn't drip anywhere. I've always said if I had an additional 3 hands, it would make injecting so much easier. In nearly all cases, unlike CT or X-ray, the patient has to wait after the injection while the radiopharmaceutical is taken up by the relevant area of the body in order for a good quality image to be formed. These are examples of some of the time delays between injection and imaging. If the patients are imaged too soon or too late after injection, the images will not be of optimal quality for reporting. Most nuclear medicine departments will have a separate waiting area for patients that have had a radioactive injection, and for PET, patients will have their own cubicle to wait in. Unlike CT and X-ray, you can see from these times that our images take quite a bit longer to form. It is important to know if a patient is claustrophobic or has difficulty lying reasonably flat for any particular length of time, or if they have any continence issues. From this slide and the previous one, I hope it shows that you can't just inject all your patients at 10 o'clock in the morning if you only have one camera to image them all on. Also, as well as thinking about how much radioactivity you have left, you have to think about the expiry time of the product. As has been mentioned by Jen and Maite, the radiopharmaceuticals are usually made very early in the morning, and their shelf life may mean that they have expired by, say, 2 o'clock that afternoon. I'm sure you can appreciate that from this slide and the previous one, getting the timings right is very important. And one other big difference from CT and X-ray, our patients are still radioactive after they finish the scan. General advice to patients will be about keeping their distance from other people. However, at the moment, everybody is doing that. For an inpatient, ward staff will be advised to keep their distance as much as possible and to store any linen or waste items in a separate area for a designated length of time before they can be disposed of, which is to allow for any possible radioactive contamination to have decreased to safe levels. Patients will be advised the time that they will stop being radioactive at. If they were traveling abroad in the days following the procedure, patients are provided with a letter detailing the injection they had in case it alarms any radiation detectors in the airport. I've talked about some images, but I haven't shown any yet. Here you can see 2 examples of a bone scan. The image on the left is a normal, regular bone scan, whereas the image on the right shows a patient with multiple bone metastases in 2 different grayscales. Bone scans are most commonly used to show breast, prostate, and lung cancer metastases. All bones are repairing themselves all the time, but when they are damaged, the repair is much more obvious and appears as a hotspot or darker patch on the scan. For the purpose of this talk, I've taken 2 very contrasting images, but you remember I mentioned my patient who fell in the bath. The image might show the only hot areas on the ribs, which are repairing from the fall. Therefore, it is important to know the patient's history so that the radiologist can accurately report the scan. A cardiac MUGA scan is used to calculate the ejection fraction of the left ventricle of the heart, or in simple terms, how well the left ventricle is squeezing to push the blood around the body. It can be used in cases where patients are having treatment, so that may damage their heart. For example, the breast cancer treatment, Herceptin. The patient is connected to an ECG, which takes the heart rate of the patient for the duration of the scan. Each heartbeat is divided into sections, which the computer is then able to add all the individual sections together to give the image on the right, which is a summary image of 1 heartbeat. Also, with the images that are required, a region can be drawn around the left ventricle to calculate how well the heart is pumping, which gives us a percentage value. In this example, it is 60%. Anything above 55% is considered normal. Patients may have this test prior to starting treatment and then at regular time intervals during their treatment and possibly afterwards as well. For example, every 3 months, to check how their heart is and to compare it to the original baseline measurement. Sentinel lymph node localization is commonly used to identify the closest lymph nodes to the tumor in breast and melanoma cancers. These lymph nodes can then be identified during surgery using a special radioactive detector, removed and sent to pathology to be examined for any metastatic cells. This technique reduces the need for all lymph nodes to be removed, and therefore this reduces the risk to the patient of developing lymphedema in the future. This is a SPECT-CT or 3D image taken of the pelvis to show the lymph nodes relevant to a melanoma on the leg. While the technique of sentinel lymph node identification is predominantly used in breast and melanoma, there are many studies investigating its application in other cancers, for example, head and neck, cervical, endometrial, and prostate. This is a PET-CT scan, which is showing the presence of a liver tumor. On the left, you can see the CT scan. In the middle, you can see the PET scan. And on the right, you can see the 2 images fused together. Combining the images really makes a difference in being able to accurately see the location of the tumor. As I mentioned before, it is important that the patient remains as still as possible for the duration of the scan, as any movement can lead to difficulty in fusing these 2 images together, and at worst, having to repeat parts or all of the scan. Here is an example of a PET-CT scan showing esophageal cancer and 2 metastases, one a lymph node and the other the left adrenal gland. It is also possible to see some uptake in the kidneys and the bladder. This is because the radiopharmaceuticals being excreted via the urinary system. It is important for patients to drink plenty of fluids and go to the toilet often. It is worth mentioning that not all areas where there is an increase in radiopharmaceutical is a bad sign on images. Depending on the properties of particular radiopharmaceuticals, they may naturally show in the heart, the brain, the liver, digestive system, or urinary system, as well as any cancerous areas. If a patient is cold after injecting, there may be uptake in the brown fat of the patient, as can be seen in the set of images on the left-hand side. And if a patient has had a particularly strenuous session at the gym the day before, then there may be quite a bit of muscle uptake, as can be seen on the image on the right. For the final part of my talk, I would like to go through some of the obstacles and things to think about when scheduling a scan for a research trial, and give 2 examples of trials that I've had to schedule scans for. Many trials that have imaging as part of the trial will require the department to undergo a qualification procedure for each camera that will be imaging patients. The comprehensiveness may depend on how much the imaging is part of the trial question and outcomes. For standard of care imaging that is being performed as part of a trial, information on the cameras that will be used may be sufficient. But if the imaging is a key part of the trial question and outcomes, a much more extensive qualification procedure may occur. This could include a visit to the site, sample QC information and images, sample patient images being sent for independent analysis by the imaging lab. I did have a trial whose qualification of the camera required a total of 2.5 days worth of imaging, which was extremely difficult to schedule around a full-time working department. Most research trials have generally 3 main parts where imaging will be required. Screening, which is after consent but before randomization or treatment, while the patient is having treatment, and during the follow-up period that the patient is monitored after having treatment. For the second 2 areas, treatment and follow-up, that's usually not too difficult to schedule your imaging at the appropriate time point. But with the screening period, it can be a little bit more tricky. And let me explain a bit more about why that is. I think you may have heard from Jen that most departments work on a 5-day week basis. And as you heard from both as well, radiopharmaceuticals have to be produced every day, which means if the radiopharmacy doesn't produce, then you can't inject and scan patients. Some radiopharmaceuticals may only be produced on certain days, as it can depend on the amount of radioactivity that is available to use. For bank holidays, what would usually happen on a Monday is then moved to a Tuesday. And this might be things like weekly maintenance of the cyclotron or weekly cleans of the radiopharmacy. These can affect the timings of the workload of radiopharmaceutical production on those Tuesdays. As I mentioned before, the patient continues to be radioactive after they have finished their scan. It is therefore advised for them not to have any other tests done afterwards on that day. If multiple nuclear medicine scans are required, there may have to be a minimum gap between them in order that the radioactivity from one does not interfere with the images of the second. For example, a cardiac MUGA and a bone scan should have an absolute minimum of 48 hours between both injections, but longer would be preferable. For radiopharmaceuticals that are required, a routine one would usually take 1 to 2 working days. However, a novel research radiopharmaceutical may take much longer than that. For example, the kit or equipment may have to be ordered in specially, as it may not be kept in stock. There is also the question of scheduling the scan itself. There may be limited capacity in the department at that particular time. Also, just as radiopharmaceuticals may be produced on certain days, this means that those corresponding scans will take place on certain days. As I showed previously, our imaging can already take some time to be performed. And if a trial has particular requirements like additional images or imaging for a longer amount of time than usual, then this may affect when it can be scheduled on the camera. Let me show an example of a trial where imaging is required. In this trial, after confirmation that the patient was eligible, the patient commenced a medication. The 2 PET-CT scans were part of the investigation to see the effects of this medication on the tumour. The PET-CT scans had to have a minimum of 8 days between each other in order to allow some time for the medication to take effect. The supply of the radiopharmaceutical would only be delivered on a Tuesday, but not in the weeks that there were bank holidays. And the treatment had to commence 28 days after consent was received. That all sounds relatively straightforward, yes? Well, not quite. Here is what that timetable would look like on a practical sense, trying to fit all the requirements into an actual plan on a calendar. In an ideal world, the scans could have been performed on days 9 and 23 of screening. However, now day 9 is after a bank holiday, which rules out having the scan on that week. Then, if the first scan took place on day 16 of screening, you can see that ideally to allow for a minimum of 8 days between each scan, the second scan would take place on day 2 of treatment. However, both scans have to be completed prior to commencing any treatment. Here is another example of a trial with nuclear medicine imaging. A bone scan and a PET-CT scan had to be performed in the screening period of this trial. The bone scan and all other eligibility tests had to be completed, and the patient confirmed as eligible prior to requesting the PET-CT. There was a minimum of 5 working days notice for the PET-CT and a minimum of 3 working days for the report to come through. And if confirmed eligible on the PET-CT, then the patient could be randomized. The trial treatment had to commence within 14 days of randomization. And remember the bone scan? Well, that had to be within 28 days of the start of treatment. If not, it would have had to be repeated. This is a timeline of all those time points that I mentioned in the previous slide. You can see that the bone scan has to be as late as possible in the screening period, but still early enough that there is time for the PET-CT scan to be completed, but still within the 28-day time frame of the proposed treatment start date. Apologies for the amount of information on this slide, but this shows how the timeline actually fits into a 5-day working week. In this case, it was really fortunate that the bone scan and the report were all completed on the same day, which allowed the eligibility to be confirmed and the PET-CT request to be sent through later that same day. Then the PET-CT was performed 5 working days later, after which the report was sent through to confirm that the patient could be randomized onto the trial. Treatment was then started 13 days after randomization, which was just within the 28-day time frame, so that the bone scan did not require repeating. So how can these obstacles be overcome? Well, preparation is key. In all the trials, the patient has to have a minimum of 24 hours to consider whether they wish to participate or not. And this time can be used to highlight to departments that there may be an upcoming patient and to speak to them about their schedules. For example, check to see if they have any planned engineering that could take a camera offline for a day or two, especially if trial scans are only performed on particular cameras in the department. Plan a patient diary to see how everything will fit together, especially if you know that there are upcoming bank holidays, which could affect the supply of radiopharmaceuticals or shorten the availability of imaging time that week. It's always good to have a backup plan and think of things that might happen. For example, if there is a radiopharmaceutical production failure, you are likely only to find out that morning, and you may need to completely rejig the plan and what is going to happen on that day. And I can definitely say that this can happen, as it has happened very recently to us here in Oxford. Once you have a plan, some departments may allow spaces to be reserved in their diary. I have found that this is always easier, and it's easier to cancel a space than to try and book one at the last minute. Where possible, coordinate tests to happen on the same day. For example, an ECG, blood tests, and a CT scan could all be done prior to having a bone scan injection and imaging. It can take a little bit of toing and froing to get it all organized, but the patient is really going to appreciate it, as it reduces multiple journeys for them, especially if they live quite a distance from the hospital. And if you know that there is a tight turnaround time for requiring the report of a scan, try and speak to the radiologist that will be reporting it, just to highlight that it is for a clinical trial. I hope you've enjoyed listening to my talk. Apologies that it was such a speedy run through of all the information, which was just the tip of the iceberg in relation to what happens in nuclear medicine and PET-CT. And I will now hand back to Elizabeth.
Okay, fantastic. Thank you so much, Edith. So we'll now move over to Dr. Daniel McGowan. So I'll provide a quick introduction as he sets up his slides. So Daniel is a principal clinical scientist working at Oxford University Hospital's NHS Foundation Trust, where he is a radioactive waste advisor and medical physics expert who leads training and research for the radioisotope physics group. Daniel has been awarded 2 personal fellowships from NIHR and has published 40 papers on a range of functional imaging, half a senior or first author. He is a co-investigator on several imaging clinical trials, including the NCITA Oxford exemplar, the FIG trial. So thank you very much, and over to you, Dan.
Can you see the slides all right?
You can see the slides, yes, thank you.
Thank you. Okay, thank you for that introduction, Elizabeth. So I've got the last talk about legislation and regulation, so it can be a fairly dry subject, so I'll make it as interesting as we can. Let me change the screen. Oh, there we go. So first of all, starting with an image, because that's obviously what we all want to do. So all the legislation I'll talk about is designed to kind of allow us to get these images that we want at the end of it. However, there's obviously radiation risks to the patient as well as to staff and public. I've been using ionizing radiation, so Maite's already discussed some of the issues around that. So we'll go into a bit more detail about the different types of legislation that there are. But essentially, nuclear medicine is a very highly regulated modality. So within just nuclear medicine, there's a whole range of different pieces of UK legislation. Some of it differs for the devolved nations, but broadly these are the items. So we have the ionizing radiation regulation, 2017. They're looked after by the HSE, Health and Safety Executive. And within your hospital or institution, you have what's called a Radiation Protection Advisor, or RPA. They're the ones that can advise you on compliance to that specific regulation. We also have the Ionizing Radiation Medical Exposure Regulations 2017, or IRMA 17. This involves advice from the medical physics expert, or MPE, and it's kind of enforced by the CQC, but it also involves RSAC, which you've heard a bit about today, and I'll talk a bit more later. And that's one of the hats that I wear for the Oxford hospitals here. There's also the environmental permitting regulations, which run to over 300 pages, or EPR 2016. And you're kind of the institution or hospitals given advice to those from your radioactive waste advisor, and they're enforced by the Environment Agency, who come in generally about once a year and inspect your hospital to check you're complying to their regulations. And all these different enforcement agencies can kind of issue notices to kind of suggest you can improve, give you suggestions, all the way through to they can kind of take you to court and fine the hospital if you aren't compliant to the regulations. So it can be bad for your bank balance if you don't comply. And lastly, if you transport radioactive material, there's the Counter Dangerous Goods Act, use of transport regulations, and for that you have the Dangerous Goods Safety Advisor, and that's regulated by the Office for Nuclear Regulation. So where do you kind of know who your different advisors are? So within your institution, they should let you know. For RPAs, RPAs and MPEs, there's this body called RPA 2000 that basically keeps a list of all the people that are allowed to act as these sorts of advisors. So you kind of click on the link and it gives you a list of all, in this case, 247 different RWAs there are within the UK. So you kind of check that you're legitimate and allowed to give advice, and it gives you your expiry date. And all of these things, they last about 5 years, then you have to renew them. So I did that, finished that last weekend. So my large portfolio, so you kind of have to have evidence of what you've done in the last 5 years for giving that advice. It gets sent off to different assessors who then decide whether or not you've done enough kind of CPD, enough professional practice to maintain that accreditation for the next 5 years. And it runs to kind of over 100 pages. It's a large document to put together, really. So within the institution, in terms of IRR 17, which kind of mainly focuses on staff exposures with lots of imaging modalities like X-ray, CT. When you're not taking that picture, there's no kind of risk, the X-ray beam isn't on. Whereas with nuclear medicine, it's a little bit different. Once you've injected that patient, they themselves are radioactive and stay radioactive. So you have to be careful why you're injecting and how you do that. But also once the patient's finished their medical imaging, when they go home, you give advice for how they can be with their whoever they might live with, and also as they go through the hospital, what precautions they have to take. So different ways that we kind of can prevent contamination. So you've seen a bit about the different things around radiopharmacies, and we're all used to with COVID times now, all different PPE, but essentially it's the same sort of thing to protect yourself against any kind of spillage of radiation, using different like a mask or gloves, typically or overshoes, depending on the area that you're in, and the risk that that particular radionuclide will determine what PPE is necessary for you. As Maite has already mentioned, the key bits are time-distance shielding within nuclear medicine. Distance is kind of the easiest one. You just take a step back, and inverse square law, you reduce it a lot. But you also use shielding as appropriate. So, for example, when we treat, Edith mentioned about molecular radiotherapy treatments, when we treat therapy patients on the ward, they're actually within lead-lined rooms. So therefore, when people go and see them to check on them, they're protected because there's lead between you and that patient to reduce the staff members' exposure. And again, if you've got a patient with radiation, the bigger your kind of distance from them, so here if you're really close, you get sort of 0.5 millisievert per hour. Go out to 2 meters, it goes down to 0.03 millisieverts per hour. So you can really reduce your exposure just by taking a step back. Another aspect to be careful with in terms of contamination is on your skin. So it's always very important to monitor yourself regularly, as already been mentioned, both when you kind of handle injections or even radioactive waste, to ensure there's nothing that happens to this poor person here where they've got some issue on the skin, they didn't realize it. And if you don't realize at the time, some of the radiation can be quite damaging to your skin. Or even if you have an accident like here, you can see this is smashed into this transport document, which then in transport box, which in theory could then leak some radiation down onto the floor here. So the way to prevent this is simply use PPE, follow the rules that are in that particular place. And if you do find any contamination, just wash it off as quickly as possible, and you can kind of reduce the time you've been contaminated by regular monitoring. You can see here, as you kind of wash your hands regularly, you kind of reduce the amount of technetium that you have on your hand. So that's about IRR. So moving on to the environmental permitting regulations. So this act regulates the accumulation, use, keeping and disposal of radioactive substances. So you have different permits, both for open sources, which are the sources you might inject into patients, as well as sealed sources, which are typically used to kind of check scanners, how they're performing, that Edith mentioned earlier. Within our permit, it specifies the exact radionuclides that you're allowed to keep on site, as well as the amount of radioactivity you're allowed to both hold and discharge and hold as waste. So there's different categories for how you can store your radioactive products. So you need to make sure that everything you're holding or ordering is within those different categories. So if you're setting up a research trial, you need to be sure that the sites you want to do that research trial on have that specific isotope on their license, because it's needed for them to be able to do those treatments, deliver that diagnostic test. There's also very strict requirements of theft and damage to sources, particularly the sealed sources, and we get inspections by the police as part of that as well. However, as I said, if you have a research trial which needs a new radionuclide, or alternatively, if it's using a lot more radioactivity than you typically would use for your normal clinical practice, then you have to do a permit variation, and this can take a lot of time to vary. You have to produce a lot of different documents, a lot of procedures have to be sent off to the EA for them to assess and determine if they consider you're safe to increase your permit limits. And as part of that, you have to do an environmental assessment. So obviously any patients that we treat in Oxford, when they go home, we'll discharge any radiation that isn't kind of attached to them, typically through their urine. So that goes out to the sewage plant. We have to keep an eye on what we discharge in Oxford, but also other private institutions or universities within Oxford that go to the same sewage plant. So a project I did many years ago, over a decade now, was looking at the radioactivity levels in the treated effluent. So after the water's been through the sewage plant treats it, spits it out into this little brook that then goes off onto a hotel nearby. So it's like the pretty little brook in the river, but it's basically just pure sewage water that's been treated. So we were monitoring with a bucket and, in this case, computer cable thrown in to collect our different water samples to see what radiation levels we were detecting in the brook. And this is part of our kind of application to the EA to allow us to treat more patients. This is our kind of proof of how we could do this. So that was kind of the initial way we did it. And then we were able to loan a much better system. So rather than me clambering on this riverbank to chucking a bucket in, we could have this device here, which was basically an automated water sampling that the EA use for kind of assessing water quality throughout the UK. And with this device, you basically, it's like telephone banking. You ring it up, say, I want you to start counting at this point, press 1 to start counting, press 2 for stopping counting. So you kind of tell it to go, and then every couple of hours it would take a sample automatically, install the bottles for us in this little tub. So then what we could do is we could compare the patients on the ward. We measured their radiation levels regularly, so we could tell when they went to the loo, and we could measure the, which is this pink line here, the patient on the ward, in this case an iodine patient. Then we measured the iodine levels within the treated effluent as it's about to leave the sewage plant into the brook. So you can see you kind of get a little spike, basically a day or so after your iodine patient enters the ward. So therefore we can kind of measure, as the patient's in the ward, they go to the loo, a day or so later you can measure that kind of spike of radiation smeared out as it leaves the sewage plant. And in terms of your kind of permit variation, you have to model all these different things. In this case, have to calculate what the sewage treatment worker would get, what the farming family, children playing a brook, angler family, all these different categories. You have to kind of prove to the EA that your increase of kind of use of radionuclides at your site won't adversely affect any of these specific groups in terms of environmental assessment. So you can see there's quite a lot of work and modelling that has to go into getting these different permit variations approved. So if you do want to add a new radionuclide or increase what you want to do, it's good to kind of talk to sites early and get this process going as soon as possible, because it takes many months to get it all together and get it approved. It also costs quite a bit. I think it's like several £1,000s, about £7,000, I think, to do a complete variation just for the EA cost. So that's EPR. Now we move on to IRMA, which is a large piece of legislation focused on patients. Came in just before Christmas in 2017. So in IRMA, the key thing is it defines 4 what's called duty holders, and they're the people responsible for implementing these regulations. So the employer has overall responsibility to ensure that all the exposures must do more good than harm. Then you've got the referrer, and they're the people that request the radiation exposures. They have to give enough detail of why that exposure is necessary to allow this practitioner in order for them to justify the exposure. And the operator is the person that actually does the exposure. And then the other duty holder involved is the medical physics expert. So we're responsible for ensuring everything's optimized and helping to write all the different documentation you need to kind of prove compliance to this legislation. So the key thing with IRMA is justification. So every use of radiation must be justified, and crucially it must do more harm than good. So it must be very low risk and very high benefit because it's used for medical purposes. That's how it's justified. As part of the kind of the MP role, we like to optimize these images. So as Maite already mentioned, we want to ensure that all the doses that we give to patients are as low as reasonably practicable or a LARP. So that can be kind of just using the equipment properly, as we've already mentioned, using right exam, right patient, and also getting the lowest, giving the lowest injection possible, lowest activity of nuclear medicine or PET to get a clinically effective image quality. Obviously, if you give the patients a huge injection, huge effective dose, you'll get stunning images, or you could scan them super quickly, but that may not be necessary. So you need ways of assessing what's needed for that particular patient, that particular indication. So typically we do part of this by phantom scanning. So this is a NEMA image quality phantom. This is what it looks like scans. You can see it's got cold lesions, hot lesions. So we use this to assess how different image reconstructions are performing, so we can kind of optimize the settings that are used on that scanner. that's kind of what's historically been used. We try and use more kind of realistic phantoms where we can. So this is a 3D printed phantom called Abdoman, made of ICR, where you have a kind of 3D printed liver, different lesions that you can insert into it. And for this particular project, we're looking at the effect of respiratory motion. So as patients breathe, so putting this on a motion platform that moves the phantom with a kind of a typical patient's respiration, allowed you kind of, you can see the liver. This is kind of a hot rim, cold necrotic center. So you can kind of really test out the imaging and find the best reconstruction settings for that particular indication, the protocol that you're setting up. And this example for PET, where this is our kind of image quality, and then we worked together with the manufacturer, we optimized it, and this is developing, correcting for motion, so you can see these lesions are much more sharper than they were in this previous one. So the application of IRMA to research exposures. So a key thing is how you define an actual research exposure. So it's any exposure required by the research protocol following initial consent from the participants. So therefore it includes all exposures carried out on participants and by the protocol, including those which would be part of routine clinical care if they weren't in the clinical study. So it's quite critical within your IRAS form to list all these different exposures out. So within IRMA, in terms of research specific, we have to ensure that the individual is informed in advance about the risk, particularly from the research exposures. So that's typically done through the patient information sheet. Also, the individuals participate voluntary, approved by a rep. They still need to be justified by an IRMA practitioner, typically a radiologist. And for nuclear medicine, often that's the RSAC practitioner, so an RSAC license holder, which we'll come on to. And some research studies also we're doing patients as normal volunteers, so then you have the practitioner will have to pay particular attention to those cases. So looking at the IRIS form itself, so there's part B, section 3 is the bit that physics would always look at. This is the key bit which determines what you're doing, both in terms of CT and nuclear medicine of your investigation. So here's an example from our FIG Oxford exemplar where it's a brain PET CT using F-DOPA. This is the activity we're recommending we give, how many administrations you give, and the effective dose. And the key is in this section, you have to include all the different nuclear medicine isotopes that you're injecting. As I said, whether they're standard of care or research only, you'll say later on which is specified as research or standard of care. And there's another part where you list all the different procedures that the patient has. These two must match. Typically, lots of, when we review kind of sites before we open with an Oxford, this is typically the problem that sometimes come up is where there's an issues with the number of things listed within A1 compared to the A19, which is all the different procedures that have to take place. So this redacted material bit and dose bit is all completed by your kind of lead MP and the lead CRE, which is clinical radiation expert. So the clinical radiation expert, they assess whether the protocol could involve any additional radiation exposure, and they advise the CI and the REC on the suitability and ethical acceptability of these additional exposures. They should be a registered medical dental professional, crucially with clinical expertise in that specific modality, be it nuclear medicine, PET, X-ray, or radiotherapy. CRE can be on the research team, and they're based in the UK because it needs knowledge of and this is again for our FIG study, what we had. So in this case, a nice short series, just saying the radiation dose from the additional PET scan is of no clinical significance to patients with Gallagher. So they take into consideration the patient group that you're scanning or treating as to the clinical impact for that specific group of people. Moving on to the MP, so we will perform what's kind of a dose risk assessment for all the different radiation exposures proposed in the protocol, and we state whether these are kind of standard of care or additional, so that the REC can easily see which what's the patient would get normally, and what by taking part of this trial, what's the additional radiation exposure they're getting. Again, it needs to be an MP that has expertise relevant to that specific procedure. And often, especially in nuclear medicine, where we have SPECT CT or PET CT, you therefore need a nuclear medicine PET MP as well as a diagnostic radiology CT MP to be involved in this risk assessment. So this is again the one for our Oxford exemplar. So you can see it's a little bit longer than the CRE. So we've made it clear that it does require exposures to ionizing radiation. This is what the CT component is. This is what the nuclear medicine component is. Equivalent to 2.3 years of average natural background radiation in the UK. And as Maite mentioned, the background radiation, like we've gone through what the different aspects that has, but also to bear in mind, say somewhere like Cornwall, the natural background there is about 7 millisieverts a year. So this is average across the whole of the UK background. Obviously, it's less than if you had a year in Cornwall. And the statement that's kind of key that goes into your patient information sheet is saying the risk of developing cancer, the consequence of taking part in the study is this percentage, tiny, 0.03%. And then you always need to add in, like, for comparison, the natural lifetime cancer incidence within the UK is about 50%. And you can see here, this is then done by, DR was done by College James, and then I did the nuclear medicine part. So often for more complex studies where there's radiotherapy as well, you have 3 different MPs listed. Only one of them will sign it off, but you all kind of are listed there, so you kind of take responsibility for your bit that you've written. So within the radiation reviews, obviously it's crucial that the MP reassessment reflects the patient pathway, and this patient information sheet needs to include similar explanation so that the patient can be fully informed about the risk of participation. And there's different guidance on the HR website about what that statement should be, and it depends partly on the prognosis of the patient groups that you're examining as to how you should phrase that radiation risk to the patient. So how do we comply with these IRMA requirements? So individuals are informed in advance about the risks from radiation exposures. This is through the patient information sheet. Participate voluntary, obviously research consent that we would do. Approved by ethics committee, IRIS form when it gets signed off. Procedures justified by own practitioner, and it's made clear when you kind of refer patients how that's done. Again, on the IRIS form, if there are kind of normal volunteers, we have that included. And within the IRIS section, we say what the dose constraints and target doses are to deliver the protocol and the doses that we've assessed. So there's different ways of doing your radiation assessment. So the kind of MP area I've just kind of shown, we've done locally through Oxford, and you can do through your own institutions. HRA have now launched kind of a national radiation insurance program. So I take part in this as well. They've got like a list of national kind of approved MPs and CREs, and we get allocated kind of different studies randomly throughout the year. And you are sent all the documents, and you have to write your MP assessment for that specific bit. The disadvantage of these is often the MP and CRE aren't necessarily in the same place. So whereas with local kind of approvals, we can kind of check with each other how we're doing it. With this, because it's kind of picked from a list, it can be fairly random. Typically, if we're asked to do one, we try and make sure at least within like nuclear medicine or the CT one, it's the same site MP. But I've been involved in a study recently where there's 3 MPs, because it's involving a lot of different modalities. All 3 of us are in different places in the UK, so it's a lot of emails back and forth to agree kind of the final statement. But this is kind of typically what they kind of push lots of people to use, and it works quite well. It's £500 plus VAT per specialty. So if you've got lots of different modalities within your trial, that can get quite expensive. Whereas typically kind of local MPs, depending on your site, when they do the assessment for you, that can be cheaper. So it depends on your trial as to what system you want to use. Another big aspect, especially for us in nuclear medicine PET, is RSAC. So for whatever trial you want to do with a nuclear medicine, you have to get RSAC approval for that use of radionuclides. So under the old regulations, it used to be there was a practitioner certificate, and they had separate certificates for diagnosis, for therapy, and for research. And for every single research trial, we used to have to send off a paper copy, wet signed, to RSAC to get back, and keeping track of all this paper and getting all the paperwork was a complete pain for everyone. So as part of IRMA 2017, this was all improved and modernized. So now there's what's termed a split license approach. So there's an employer license and a practitioner license. So your employer license allows us to do it at specific sites. So in Oxford's case, at the JR or the Churchill Hospital sites. So there's a list of the radionuclides that we're allowed to inject and treat people with on each site. As part of the kind of the application process, they check the staffing that's available, the experience at the site, and what equipment you've got. Then there's a separate license. The practitioner has a license, and their single license will cover them for diagnostic therapy and research. And the license stays with that practitioner. So if a practitioner works at a couple of different hospitals, they don't need a new license for every hospital, which is what we needed before. Both of these are valid for 5 years. You still have the sponsor PRA approval. So for each trial, the sponsor will get an RSEC approval letter, license, which can then be sent to the employer and practitioner for that each site individually for them to kind of see that RSEC have agreed it. And this is typically what they look like. So when they've been kind of authorized, when it expires, who did it, and it lists a whole long list of procedure codes, the radionuclides, what you're allowed to do, what the indication is, and crucially here, whether it's authorized for research. It may be you've got certain procedure codes that you don't have for research. Most institutions will have anything they can do diagnostically, they'd want to do research-wise as well. But it means it's much easier if you've got a research trial that comes up, whereas in the past we used to have to always do an application to RSAC to get a new certificate for that specific research trial. Now if there's a new research trial that comes on, often they involve FDG PET, bone scans, GFRs, markers. Those are kind of the 4 biggest ones that are used for all research trials. But if our practitioner already has it authorized for research and it's on our site license authorized for research, we don't need locally any additional RSAC license. You wouldn't still need the overall study RSAC approval, but you don't need any specific license for that radionuclide. Where you do need to apply to RSAC is if it's kind of a novel radionuclide or radionuclide that you haven't used before on your own site, then you have to apply to RSAC to have it on your employer license and your practitioner license. Updating practitioner license with new procedure codes is free. Updating your employer license for new procedure codes you have to pay for. So the trick is if you know what trials are coming up, you can do as many kind of updates as possible in one employer application to kind of save us up a bit of money. So with that, thanks for listening, and any questions?
Thank you so much, Daniel. So if we move on to the Q&A, there's a couple of questions in the chat, so thank you to those who have posted. So there's one here around if the CRE advises the REC regarding ethics of the exposure, but they can also be on the research team, is there a potential conflict of interest there? And if so, how is that managed?
If the CRE, you said?
Yeah, if the CRE advises the RE ethics of the exposure, but they can also be on the research team.
So that's the official guidance, is that they can write the CRE, and so obviously when the REC examine it, they'll see if the CRE is also involved in the research team. And like typically you wouldn't have it as the CI of the trial would write it, but because lots of these, especially if it's a really novel agent, that maybe people don't have the expertise to kind of give all the details, so you can be limited to who you can use as a CRE. But that's the official guidance, is you're allowed to use a CRE part of the research team.
Perfect. Okay, thank you very much. Another question from Ruth. So do you foresee a move in the future to make it a requirement to use the central radiation assurance system?
I think they're trying to, but at the moment there's not enough central reviewers approved to cope with all of the different like UK trials that are going on. But I think that would in a way be simpler both for REC and for people, because then you've got one kind of streamlined process, you know how everything's got through and everyone's writes it in a standardized way. But at least in the short term, there'll still be local sites doing their own approvals, which you are allowed to do. But HUA are kind of actually encouraging people to use the central method where they can.
Great. Okay, thank you very much. One question for Edith. This is from Penny. So who is it within the department that schedules the scans? So who's the best point of contact when organising trial scans?
Every department is different. So all the different departments that I've worked in between Barts and Lincoln, Guildford, and now here in Oxford, they all have a slightly different way of doing it. So sometimes, so say here in Oxford, the PET-CT scans, we actually are very lucky to have a bookings team who are really experienced in advising our patients about the preparation that they need to do. So I would go to the bookings team directly to ask, have you got a particular space available? Whereas in other departments I've had, it may be, say, the senior technologist or radiographer who would be the main point of contact to say whether it's okay to schedule on that particular day a bone scan or a MUGA or something like that. Also in some departments, for routine research tests, if you could say like the bone scan and the MUGA, they may have a policy where they keep maybe 1 or 2 slots free per week. I know in Guildford they did do that because of the amount of trials that are asking for bone scans and MUGAs. So they would routinely have, like, say, the Wednesday 3 o'clock bone scan, that's always available for a research trial. So you would have, they would have that available up until, say, maybe Monday morning. And if it's not booked, well, then they will use it for routine clinical use rather than a research trial. So each department is slightly different, but it's usually either the bookings team, if you're lucky to have some, or a department secretary or a senior technologist.
Perfect. Thank you very much. If there are any other questions, please do post on the chat, unless anyone would like to unmute and ask a question. I'll give it a couple of seconds. Okay, so I have one question, and Edith, I think this one's for you, but when checking levels of radioactive exposure, how does the finger monitor actually work? And what would happen if a staff member did have radioactive exposure?
Well, I think I'll ask Dan to help out on this one as well. But the finger monitors, they're not a sort of a live reading. The ones that I showed anyway, there are some that are available that you get live reading as you're sort of walking towards the patient, as you're injecting, as you're drawing up the injection, you can get a live reading. But those particular ones we would have for 1 month, and then they would be sent back off to be analysed. So it's usually, there's a small, it's like a crystal material, and it changes with the amount of radiation that it's exposed to. And then the laboratories that they're sent back to, they would analyse how much radiation exposure you have. So you don't always find out until sometime after. It's worth, if you can, to make a note of, say, if there was a significant spill, or if something happened to patients that you needed to be beside the patient for a prolonged period of time. For example, say they fainted when you went to inject them and you needed to be beside them for maybe half an hour or something like that. It is worth making a note so that, 3 weeks later, when you're asked, well, what happened to your finger doses, you can turn around and say, well, this particular patient, was ill and I needed to be with them for a long period of time. So there is that, so you don't get live recordings with those particular monitors. Dan, do you want to add anything?
No, that's right. And then the advantage you get is once you've got it measured, if there was a high exposure and you had to investigate for whatever reason, the company can tell you if it's kind of a one-off high exposure on a specific event, like Edith says, like a spill, or if it's been kind of continuously, you've had slightly high for the whole month, so you can then guide your investigation appropriately. And there's if it was high above investigation levels, there would be a kind of a thorough investigation to see what happened, to kind of an action plan to address it for the future.
Especially for new starters, you would always expect their finger doses to be a bit high as they get used to handling radioactivity, drawing up, because, as I said, when you're wearing them and a set of like normal latex gloves or whatever, gloves on top of them, your dexterity and the feeling in your fingers is so different. So it takes quite a bit of practice to get used to that.
Sorry, can I just add to that? So generally, from the chemistry point of view, whenever we get our readings back, so what generally happens is if an operator has like high finger dose readings, generally for the next few months, you are restricted in the number of operations you can do. So instead of you being the operator performing certain procedures in the radiochemistry lab, so it'll have to be somebody else in order to ensure that your annual doses are within the limit. So if the dose of one particular month is over the limit, then yes, the number of operations you will be performing in the upcoming months will be reduced.
Great, that's really interesting, thank you. Okay, so I think that might be all the questions that everyone has. So I think we're coming to a close for the workshop. So I'd like to thank you very much to the speakers for presenting and providing such a fantastic insight into nuclear medicine, into oncology. So thank you, Maite, Jen, Edith, and Daniel. Just a note from me that the event has been recorded and that will be made available on the NCITA website and also on the NCITA YouTube channel, and we will provide a link to that. And I'd just like to thank you very much for joining and enjoy the rest of your day. Thank you.